Hello and welcome to my channel, Sam Biology Class. Friends, today we will discuss about the principle and applications of transmission electron microscope. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Transmission electron microscopy is a high-resolution imaging technique used to study the detailed internal structure of different types of cells, intracellular organelles, to identify virus, to study the mechanism of various diseases at the cellular level, to characterize nanomaterials and particles, and also to examine metal, alloys, ores, and crystals to identify the defects. The instrument which utilizes this technique is known as transmission electron microscope, TEM. The principle of TEM is very much similar to that of a traditional bright field light microscope. In both of them, radiation is transmitted through the specimen, forming a detailed two-dimensional magnified image. In addition to this, both of them use lenses to focus the radiation and to magnify the image. However, there are differences. Unlike light microscope, which uses glass lenses, electron microscope utilizes a series of electromagnetic lenses to focus and to magnify. In addition to this, in case of light microscope, visible light is used to illuminate the sample. By contrast, in electron microscope, a fine beam of fast-moving electrons is used for illumination. The first transmission electron microscope was discovered and developed by German physicist Ernst Röska and electrical engineer Max Knoll in 1931. For this significant work, Röska was awarded the Nobel Prize in Physics in 1986. This figure illustrates the principle and key components of a transmission electron microscope. The components are an electron gun, anode, and different types of electromagnetic lenses such as condenser, objective, aperture, and projector. In addition to this, other components of a TEM are a fluorescent screen and a suitable camera for recording the image. All these components are placed vertically in a vacuum column. Vacuum condition is necessary to prevent collision between electrons and air molecules. As already said, in place of glass lenses, electron microscope utilizes a series of electromagnetic lenses that utilizes the magnetic field to control the beam of electrons. First component is an electron gun which generates electrons. These electrons are then accelerated by applying a massive voltage, typically between 80 and 300 kilovolt, between the gun and the anode. These red lines indicate the electron beam. The function of the anode is to attract the electrons, accelerate and shape them into a fine beam, which is then focused onto the specimen by the electromagnetic condenser lens. For TEM, the specimen should be ultra thin, which is placed on a support grid. When the electron beam strikes the specimen, denser areas scatter more electrons, creating contrast. On the other hand, thinner areas of the specimen allow more electrons to pass through. These areas appear bright in the image. This image is further magnified by the projector lens to form the final image, which is then focused onto a fluorescent screen. Out of different components, one is the aperture, which is located between objective and the projection lens. The function of the aperture, which is a precisely drilled metal disc, its function is to physically block stray, scattered and off-axis electron, thereby controlling the electron beam and enhancing the image contrast. As the transmitted electrons which pass through the specimen are collected by the objective, transmission electron microscope is used to study the detailed internal structure of different types of samples. Transmission electron microscope can magnify the specimen up to 50 million times. As the wavelength of electron is more than 10 to power 5 times smaller than that of visible light, the resolution of TEM is very high, about 0.1 nanometer. Because of this, by transmitting a beam of fast-moving electrons through the specimen, TEM reveals nanoscale, molecular and atomic level details of different specimens. Specimen preparation for transmission electron microscope. For TEM, the specimen should be thin so that the electrons will be able to pass through. Usually the specimen is embedded in resins and cut into ultra-thin slices by ultramicrotome. In order to enhance the contrast, the sample can also be stained with heavy metal salts such as uranyl acetate or lead acetate. After cutting by ultramicrotome, the slice is placed on a support grid and is examined under the microscope. Image formation. As already said, in TEM, a beam of fast-moving electrons is transmitted through an ultra-thin specimen. When the electron beam strikes the specimen, denser areas of the specimen scatter more electrons, and those electrons do not reach the objective. As a result, such areas appear dark in the image. On the other hand, thinner areas which are less dense allow more electrons to pass through. As a result, these areas appear bright in the image. The electrons which pass through the sample are then collected by the objective, which forms the intermediate magnified image. So the image formed by electron microscope is naturally black and white. The intermediate magnified image formed by the objective is further magnified by the projectional lens to form the final magnified image. This final image is focused onto a fluorescent screen or can be connected to a camera to develop the photograph, which is clearer, sharper and more detailed. The photograph of an electron microscope image is known as electron micrograph. So in this way, images are formed in transmission electron microscope. This figure is a transmission electron micrograph which reveals the detailed internal structure of a specimen. Applications. Transmission electron microscope has several applications. For example, in biological and medical sciences, TEM is used to study and identify virus such as SARS-CoV-2 and bacterial flagella and plasmids. In addition to this, in cell biology, TM is used to visualize the ultrastructure of cells, cell membrane, and various intracellular organelles. Finally, this technique is also used to study the mechanism of various diseases such as specific kidney disorders, lysosomal storage diseases, and muscle and nerve disorders. In addition to this, transmission electron microscopy has also applications in material science and nanotechnology, in which this instrument is used to characterize nanomaterials and particles at molecular and atomic number. TEM is also used to examine metals, ores, alloys, and crystals to identify defects and to examine different samples such as soil and rock to study their chemical composition. Friends, this is all about transmission electron microscope. For more updates, subscribe my YouTube channel, Sam Biology Class.